All right. So, thank you everybody for coming to this introductory webinar on the clinical trials methodology course. Our website is neurotrials.training, and I am Will Moyer. I am Assistant Professor of Emergency Medicine and Neurology here at the University of Michigan. I will introduce all the other course directors as well on one of the subsequent slides, but currently in attendance we have Dr. Roger Lewis from Harbor UCLA Medical Center in Los Angeles. So to talk about what we are going to cover in this discussion today, and it may not take the whole hour, is I'm going to describe some of the functions of Adobe Connect. This will be our platform for webinars, but not small groups. We'll use something called Blue Jeans for small groups, which is similar to Skype in some respects. I'm going to give you an overall overview of the clinical trials methodology course, who the people are, what the aims of the course are, the timelines, and the expectations. In addition, give a little bit of an opportunity for questions regarding what isn't yet clear about the course. I know I haven't provided a lot of stuff to you in terms of the syllabus for the year, and this will be a high-level overview, and we'll be providing you with a more specific document likely early next week after we just put the finishing touches on it, and then we will adjourn. So this is the example of the audience response system. You're going to see that the room is going to transform here. And you can see that there is a little poll over here on the left. And you should be able to vote on what your favorite college football team is. I know it was recently March Madness, and oh, I didn't let you vote. Now you should start being able to vote. The poll is now open. But I'm a big college football fan, and people are, and basically we'll let people continue to vote here. All right. And this, so when we do webinars, we're encouraging the presenters to use audience response for a couple questions like this to see these things. So then basically, after a number of votes have come in, we see who has the highest number of votes. And right now, none of the above is the most popular team here, followed by The Ohio State University. Even though I am faculty at the University of Michigan, I grew up in Ohio and went to Ohio State for college. So my favorite college football team is The Ohio State University. So anyway, that is the that is how the audience response will work. So we'll move back to the regular presentation room. The other major function that we use, there is the ability for you to raise your hand. We're not necessarily going to use that function too much in the way we do this. If you have questions, feel free to type them into the chat box at any time. Oh, I forgot I had 2 straight questions. All right, so this is just a more useful question to see like what time it is for people. So we'll flip back to this. All right, and we see that most of the people who are on the call today who are responding are here in the Eastern or Central time zones. So that is not surprising. So we will move on to the meat of the presentation. Oops. So one of the first things we said we'd talk about was the people who were involved. Like I said, I am Will Moyer. I am from the University of Michigan. I am both trained as a stroke. I did a stroke neurology fellowship, but my primary specialty is in emergency medicine. Roger Lewis, who is an emergency physician, who has advanced training in biophysics and biostatistics. Lori Gutman, who is a neuromuscular neurologist at the University of Iowa. Robin Conwit, who is at the National Institutes of Neurologic Disorders and Stroke, who is also a neuromuscular neurologist. And Chris Coffey, who is a biostatistician at the University of Iowa. One of the sort of key functions and key sort of philosophies of our course is that early involvement and collaboration between biostatistics and clinical medicine is essential to the good design of clinical trials. So what our course is aiming to do is of relevance to you. And while I'm not necessarily a fan of reading slides, I think it's worth going over these just in a little bit of detail so that you understand what the philosophy of our course is and what we're trying to accomplish for you. But basically, the goal is to identify promising junior investigators, or not always necessarily junior, but at least new to the idea of clinical trials and conducting interventional clinical trials in the clinical sort of. Oh, so it looks like Dr. Meader is having some connectivity issues. Maybe if everybody else could opine as to whether I sound okay. Just I would hate to be continuing to talk if things were all scrambled for everybody else. Okay, so there's some intermittent connection stuff. So I apologize for that. I believe I'm on a pretty good connection here. We will be able to have a recording of this that will be uploaded and should be in a good sort of format for this overall. So we have sort of varied connections across some of the people. So again, if this is so as to look a little bit uninterpretable, we will, we'll definitely be able to get you guys to watch the video later. So sorry if you're having connection issues, but it seems like it's sporadic and may have something to do with the connections that you're on, and I apologize for that. So the idea is then we've done the identification, you're here, and we want to provide you with a rigorous foundation in the design, funding, conduct, monitoring, and oversight, ethical performance, and reporting of patient-oriented clinical research, with an emphasis on clinical trials that involve the delivery of some intervention, be it a drug, a device, or behavioral intervention on patients or involved in patient care. In addition, we want to promote the ongoing professional career development by supporting you before, during, and after the program to help you follow through on your plans to do the clinical trials-based research. And importantly, the idea is that we are going to enhance the pipeline of scientifically sound, well-designed early-phase clinical investigations that will really provide the foundation for the pivotal confirmatory trials to reduce the burden of high-impact neurological diseases going forward. So we want the trials that you design and get funded to be things that are really scientifically useful and help to propel science forward. This course fundamentally addresses a gap, and one of the things that, in terms of our general philosophy and motivation for doing this, is that the early clinical trials are really amongst the hardest. And Chris Coffey will talk about this a little bit more in his talks. But even though they're the hardest, sometimes we spend the least time carefully planning them. And specific expertise in designing trials in the neurosciences is not evenly distributed across the country. And the idea is that both biostatisticians and clinicians that have experience in this area are a great resource and will help you design trials that will be more likely to be scientifically meaningful and, in addition, doable. as we've had past experiences before about making sure that the trials are meaningful and doable. And the goal of every trial should be that it can lead to a scientifically interesting result that informs future scientific steps. This is in contrast somewhat to a general goal of developing a early phase clinical trial where not much learning occurs, and it's going to basically lead to additional trials no matter what. You really want to use this opportunity with your patients and the volunteers who are acting in your research to do as much as you can to learn something that gives you the ability to scientifically decide if and how you should go forward in that with that intervention. And that's a large part of what we try to do when we design trials in this course. So one thing that I want to just sort of throw out there so it's sort of clear as to how we're going to progress through the development process for the year is to just make this definition. And my wife, who's a dietitian who does research, said that that looks like a very basic definition. And perhaps these definitions are basic, but let's just, I'll just tell you what I'm saying here. And that a protocol is a document that describes in detail how to conduct and interpret a scientific experiment. A proposal is a document that motivates an entity with money to provide grantees with the funds to conduct the research. So the idea here is that you really can't do many research trials without money or funds. There are some that you could, but there has to be some resource that's put in, whether it's your time or the time of your people in your department. And as such, sometimes the proposal is done first. The philosophy of our course is that you really want to spell out in a great deal of detail how exactly you're going to conduct the experiment, and that is the important first step. And it is actually easier in the long run to derive your proposal from your protocol than it is to do it in the opposite direction, because you will find that you forgot things if you only put them into the limited space that is conferred when writing a proposal for grant funding. And the idea is that this is time well spent in terms of developing the very specific things that you would need to do so that somebody could reproduce your research trial. That if somebody was handed your protocol, they could figure out exactly what you did, what decisions you made, how the patients were approached, how the patients were handled when they were in the trial, and really, the idea of if this was a preclinical experiment or a physics experiment, that somebody else could look at your protocol and really know exactly what you're going to do. So that is the philosophy of the course, that we spend a bulk of the time up front to get the protocol right, and then we will spend some time after the course in July where we're going to help you develop your proposal so that you can get it, you can get that funded and done potentially. So this sort of brings to this chicken or the egg. What is the important logistical consideration in terms of clinical trial planning is getting the resources to do the clinical trial. This has been one of the general challenges in neuroscience research, is that when you're planning these early phase clinical trials, you may have resources through your own institution, maybe your CTSA, but those are a general resource, and they may not have the expertise in your disease area or in neurological trials in general. A key distinction between many neurological trials, although not all, and oncology trials is the correlation of toxicity and the outcomes. In stroke, the area which I can speak most of, the primary toxicity of a reperfusion trial would be intracranial hemorrhage following thrombolysis or other intervention. Of course, that is extremely correlated with the final outcome of disability. If you're doing an oncology trial, however, for chemotherapy, nausea, vomiting, tolerability of the treatment are major factors in terms of toxicity, and they are not directly correlated with the overall outcome. Although if you can't be on the treatment anymore, then the treatment can't benefit you. So there is that indirect correlation. So the idea of this course is that we get you with people who've been there before from both biostatistics and the clinical neurosciences, who understand some of these nuances so that you don't have to go to people and resources at your own institution who are more generalists in this. And that's not to say that there isn't value in helping in working and collaborating with people locally. The idea is to help you get your ideas tuned up here so that they are more likely to lead to those scientifically interesting answers that can propel the field forward. All right, so here's another little question. Move us over to the question and answer. All right, so we have a good distribution here. Neurology is a large proportion, but we have representatives from my other fields of psychiatry and psychology, neurosurgery, and emergency medicine, my primary field. So that is good. All right. Let's move on. So the key deliverables from this course, from the perspective of the things that we want you to be getting done and sort of walking away from this course with, as opposed to the things that can sort of never be taken away, what you will have learned in the didactics, what you will have learned by doing things, is that really we want a clinical trial protocol finalized in July. And ideally, it would be excellent if that was done. It may not be its total final version, but it at least has every sort of conceivable blank filled in by July. You may have some areas where you are actively tweaking, but something that would be in a state that would be acceptable to go in as an IRB application or potentially to the FDA if that was something that was necessary. So getting that protocol to the point where it is ready with perhaps minor adjustments to be the scientific document that somebody could use to conduct the research study that you want to conduct. And then at some point in the fall, a funding proposal. And people may have other deadlines and they may be getting funding proposals in before or after that, or they may have already applied for some funding and may be getting funding before or after that, and that's okay. The goal of the course is to help you develop a proposal, but if you've already secured a way of conducting your research that you are designing within this course, that is a good thing and not a problem. So there are other things that we can help work on you with, or help you consider what your proposal might look like for the big trial that would be based on the results of the one that you're fine-tuning here in this course. So these are the key things that we are wanting you to sort of create the work product as part of this course. And that is something that we're going to be putting a lot of time and effort into making sure you're on track to do with the timeline. So the timeline here is sort of parsed out into the time periods of protocol development and then, to a certain extent, the proposal development that will occur after that initial portion of the course. So in April, it's a busy time with spring breaks and the American Academy of Neurology, which I know everybody is not a neurologist, but is sort of a very big meeting that many people are involved at. So mostly what we will be having you do in April will be the work that would be done on your own as we get some of the other organization pieces into place. Thank you for everybody who has turned in their general availability for small groups. We're working on doing the matchmaking with small group leaders right now. Then the other thing is instruments. There are some survey type data collection instruments that we use to learn about where you are with clinical research in general and also learn a bit about your knowledge base in terms of methodology and statistics in clinical trials. There is nothing, this isn't to do an evaluation of you, it is to do an evaluation of our course and see if we're changing these metrics within individual. So it's important that you do these, and we will be circulating those very soon. And I'll go into a little bit more detail on what all of those instruments are, just so you know, because many of them have names that look perhaps a little similar. So you want to know that each of them are done. And one of our sort of key sort of citizenship things going forward for the year is getting those done and getting them done in a timely fashion. So we know that there will be a lot, but hopefully you get a sense of them before too much of the other stuff in the course starts to pick up. Then from May through very early July, we will have webinars. The webinar, the sort of definition of webinar is something where there is individuals or a couple of individuals, perhaps a panel, who are describing their expertise in an area to provide you with information with some limited question taking from the audience, this more didactic. This is in contrast with small group meetings where there will be 2 faculty members along with 4 of the trainees in each group, and that will be much more hands-on and based on the items that are going into your protocol development. And those meetings will obviously occur, we have 8 small groups, and those meetings will occur at times that are convenient for those small groups, and we'll help those small groups kind of do some matchmaking to find those times. And there will be times where you are presenting, and there will be times when the others in the group are presenting. And we are going to try to come up with disease and thematically coherent groups as best we can based on the schedule demands, so that hopefully you have some other people from the areas of your study in the groups with you, so you can provide some peer mentorship to each other as well. Then, and I probably should have said July 12th through 16th, we're going to have the residential course in Ann Arbor. We're going to have a kickoff dinner on that Sunday evening, and we're inviting some representatives from foundations and patient-specific groups, such as the Parkinson's Disease Foundation, to talk a little bit about how they involve their patients and families in the research planning process, and also how they help select and fund and design research, as we believe these are important avenues for early phase clinical investigations and things and groups that we really think it will be important for you to partner with. So this will give you the opportunity to meet some of them. We're working on getting the final lineup for that. Within the course, we will have both lectures where there are specific topics that will be covered, such as DSMBs, additional small group meetings to help further refine your protocol, and then some workshops that are dealing on specific questions like clinical trial budgets or clinical trial schedules of events. And the goal of this meeting is that this is really going to be a working meeting where the overall goal is to be getting that protocol done and as good as possible, with a really tremendous opportunity to be in the same hotel with a lot of experts in the field to really bounce ideas off people and really get the thing fine-tuned. And the goal, and that is really the goal there, that we want this to be an intense experience there, and we want you to make a great deal of progress on getting that protocol to the finish line. Then after that, we hope to quickly move into the proposal development phase, with one exception. The mock IRB is sort of geared to providing an evaluation of the protocol that you would be hopefully finalizing in Ann Arbor, with the other elements of the small groups targeted more toward, and also webinars during this time period, targeted more towards how to communicate a good proposal that would motivate funding agencies to provide support for your project. So that's the general timeline of what's going to occur over the course of the year. The instruments that I talked about in terms of when we assess these, there's a demographic survey. You already did a demographic survey, but we did that anonymously based on all participants. So we'll administer something that's very similar so that we can get a sense as to if our participants who we selected for the course differ from the overall universe of people who applied for our course. So that will be something you'll be receiving soon. Obviously, that would be something that's very quick to fill out. There's also a self-assessment that was developed by the CTSA network that basically gives you a number of questions where you're asked to basically rate yourself on how confident you are in doing various clinical research tasks. And some of these things are things that are addressed by our course, and some of them are not, and that's okay. We're not going to teach you how to do qualitative research, although that is one of the things that's on the CRAI because it's a general instrument about clinical research. Then, in addition, one of the other items will be a methodology, statistics, and clinical trials knowledge quiz. You'll get one version of this before the course and one version of this in the fall. Course evaluations, I don't have a link for this, as this you could evaluate me on this, but this is generally an information session. But once we get into the content-based webinars and other sessions, we'll have course evaluations for each of the sessions. And then something called an outcome form. And when we talk about outcomes here, we're talking about your outcomes in terms of, have you gotten your protocol done? Have you gotten it submitted? Have you gotten it funded? Have you gotten patients enrolled? And this will be something that we'll submit to, that you will fill out monthly starting in July, and you'll get survey reminders for these. One analogy that we use is that our course in and of itself is a little bit like a clinical trial, although we're not randomizing people to any different groups. And that as the participants, we want to keep you, we've recruited you, we want to keep you engaged, we want to make sure that you successfully complete all of the treatments that we're providing you, or the interventions, the educational interventions, and that the final outcome is your success, and that's measured in both short-term and longer-term ways. We won't survey you monthly forever. They will sort of de-escalate to quarterly and perhaps annually over the course of our grant, but we do want to see what's going on with you over time and what's happening with the trajectory of your career. So the instruments are a key part of how we measure the effectiveness of our course. Similar to the visits after a treatment, in a clinical trial, you have to visit, you have to have your patients visit with you afterwards so you can see how they're doing. So broadly speaking, the webinars and the didactics are sort of broke up into these 4 separate but complementary themes of asking good questions in the scientific method, very important for the initial setup of your trials. Biostatistics and design considerations, how do you design early phase trials to both be meaningful and useful. Trial logistical considerations, how you make a budget, how you work on really implementing a clinical trial in a way that is going to actually work. An example of that was in the course last year, we had developed an idea for pressors after stroke in certain people with small vessel occlusions or large vessel occlusions. And at one point, there was a decision that perhaps the treatment group would go to the ICU for pressors, and the control group would stay on the floor because there wasn't funds for ICU level care for everybody in the trial. Important logistical consideration, because fundamentally, scientifically, comparing a bunch of people who were sent to the ICU versus sent to the floor for their strokes is problematic and is an intervention in and of itself. So that was something where really getting the input of people who've done trials like this before to say, yeah, we know that you can't pay for ICU care for your control group. That means you have to do something where your control group and your intervention group are getting the exact level of monitoring and other care across both groups. So it resulted in a little bit of a change in the design and intervention, which is good. And that's the idea of thinking about how, if one was actually doing the trial, what would be the potential hurdles. Responsible research, obviously, is all a requirement at our institutions to have IRB review and to know a lot about the how to do responsible research. The idea here is to add in things that are of particular interest for neuroscience trials and clinical trials in general, whereas the general training in clinical research perhaps doesn't go into as much detail about adverse event reporting and the setting up of DSMBs if necessary, and also some of the motivation for how IRBs work and how they are tasked with evaluating research. So that is another sort of major topical area, and our course is set up so that our webinars and our residential course presentations are organized in those areas. So this is the spring webinar session, although obviously it sort of extends a little bit into summer, where we are going to, these are things that, again, you're encouraged to be at. They will be like this. These are open actually to everybody. This session here is a little closed, but these are purely for the public. We broadcast them sort of to the distribution lists of NET and NeuroNext and the StrokeNet as the major NINDS funded clinical trials networks. And these are also going to be recorded and posted to our website. All the ones that we did last year are similarly recorded and posted to the website, and we may have a couple of those that we are going to assign as potential things to read for you, or not read, but to review prior to the course in April. So this is the lineup. We pretty much confirmed on all this. They're all on Fridays except for one of them, and the one on May 21st. And these should be good sessions that will help you in terms of the initial planning process. Michael Shai is going to talk about approaching industry and patient groups for partnerships. This is the idea, especially in the early phase trials. If you want to do a trial where there is, perhaps a pharmaceutical company donates drug or donates a device, how do you go about doing that? Because it's not something you want to be, it takes some lead time, and they're not always going to say yes, and it sort of depends on the circumstances. So that should be a good talk. What information you need to get started writing your protocol. Again, an early session. Sharon Yates, who's a biostatistician at MUSC and works with us on NET and also on StrokeNet, and Karen Johnston, who is the chair of neurology at the University of Virginia, are great clinical trialists. And the idea here is, what is all that information you need to get together when you're going to be starting to write that protocol? Then in June 12th, some of the high-level staff from NeuroNext are going to be talking about this from the perspective of the people who need to make sure the trial gets done, and that should be a good talk. As well, they'll all be great talks, but that will be very good. Chris Coffey will give his talk on designing early-phase clinical trials and the challenges that you have in this space. And then, while it sounds like it may not be the most exciting thing in the world, adverse event monitoring clinical trials is very important. Thinking about how you're measuring the benefit between the balance between benefit and risk in clinical trials is hugely important. It's non-intuitive, and it's not necessarily well addressed in the general clinical research training that is frequently given before. And there are even special particular nuances that are applicable to the clinical neurosciences. So the small groups are really one of the core features of our course, and they're led by core faculty teams, which is either one or two clinicians and a biostatistician with experience in the planning, conduct, and analysis of trials in neurosciences. And this basically is leading to the further development of your ideas from the application that you presented. And we know that sometimes people have new developments, and if when we're starting these small groups up, if you're like, I need to do something slightly different, it's okay. That's part of the development process. You may not have proposed exactly the right clinical trial, and new data may have come to light since you applied. So the idea here is the development of the ideas from your application, and then basically getting you to the point where you have a clinical trial synopsis that is pretty, this is sort of a 2 to 3 page overview of what's really happening in your trial prior to getting to Ann Arbor, and then hopefully the real bones of the full trial protocol in your hand there as well, so that you could be basically just updating it with what you learn and what and maybe it has a lot of comments and questions as to this I need to figure out. I need more information about sample size, and that's okay. But really to have the bones of it when you're coming to Ann Arbor and have it really well fleshed out when you're leaving. Within the small groups, and we really hope to be having some coherent groups, we want you to try to get, and we're going to try to build in ways for you to do peer mentorship, where if the assignment's due, perhaps it's sent to one of your buddies, and I guess we'll sort of have to preordain buddiness, but that somebody else from your group is somebody who you have look over your submissions for your sort of homework assignments, and you look over theirs and provide some feedback, and you do some reflection on how your outcomes look, and that we think that will make the small group sessions with the core faculty go even more efficiently, so that we can have time. And one of the expectations is that these things will be turned in advance of these meetings, so the core faculty will have time to review them, so that they can be, so that the meetings themselves can be more efficient. And when I provide the kind of protocol / syllabus to you, we'll have sort of metrics for how that's going to be operationalized in practice, how you're going to, what the due dates are going to be, and when those meetings are going to be. And again, each small group is going to set up the times of its meetings soon, once we get that all fleshed out. So here, here's just a basic overview, and I'm not going to go into a ton of detail on this about what you're going to accomplish in what we hope are about 4 small group sessions over the course of the spring. The first thing we want you to do is what we would call an elevator pitch, and this would not require any preparation of slides or basically any written document. The idea here is, can you talk about the idea in the disease area that you are wanting to study and provide motivation to the others in the group and your faculty as to why it's an important area to study, and the general scientific area where learning is needed and where you need to design a trial. Then moving on to what outcome measures you're picking for the trial. And again, the outcome measures are important because they both have to be what is reasonable for the disease that you're looking at, but also to have some resolution within the sample size that you're proposing that can provide quantitative information about whether there's an effect of your treatment, and what your design ought to be. And then that sort of pulls into the hypotheses that you would have for your trial. And then the clinical trial summary would be the short document we would give you a template of. There's one on the website already, although it may not be as easy to find as we had it within the sort of small group assignments for last year. But we'll make sure those are all in very apparent places and linked from the protocol. And then basically be working on a draft of your full protocol prior to coming to Michigan. So that's the goal of the small group sessions for the spring. So the residential meeting is 4 days. We'll be getting done by about noon on the Thursday, and we're going to have something on the Sunday evening. The expenses will be paid. If you have something in the budget for something, like for conference funding from a KT or F grant, we can talk to you about that on an individual basis. That's one of the things that's written into our grant. So we can work with you on that. If you have a K but you don't have budgeted conference funding, that's okay too. Again, something we can sort out on an individual basis. It should not cause any stress. We'll just have to see where things are with that. There'll be a combination of both the small group meetings where you'll meet with the people you've met sort of virtually in person, and then we'll have some larger group lecture type activities. Some of the small group activities will be focused on the development of your trial idea, and some of it will be on other exercises to improve your skills in coming up with a budget or a schedule of events. It'd be great and we're going to be helping you with the scheduling of travel very soon. I may have put up there that we may start working on that in May. We may start working on that a little earlier, depending on what all we have done. Ann Arbor is about 30 minutes from the Detroit Metro Airport. And if any of you are driving here, it's on the south side of Ann Arbor near the mall. And I will say that yes, we are in a hotel near the mall. So as I said before, the goal is for you to be kind of cloistered here and making a ton of progress on this. We certainly will have a night out on the town for one of the nights where we're going to have a dinner in the sort of quote unquote real city of Ann Arbor. But a lot of the other time, we're going to really be focusing on you getting work done there and using the opportunity to meet with the course faculty who are both in your small group and outside of your small group. So we'll be working on making sure dinners are there for you so that this can really be a major sort of retreat and very productive time for you. This is a general overview that sort of talks about each of the little courses. Some of the questions and some of the sessions may change a little bit for this year, and we're working on finalizing our syllabus. But the idea of some things are going on from our general ideas of the responsible research, asking good questions, and biostatistics and design considerations. And that just sort of sets up our course into its 4 thematic areas. And then in the fall, these small group sessions will probably be a little longer, where we talk about the specifics aims page, and then give some feedback on those, research strategy, and some additional webinars, perhaps on FDA issues and other things, depending on, after we walk out of the residential course, what other areas of interest we need to cover. We'll also be doing mock IRBs and mock study sections. We hope to have your involvement in the review of your own protocol so you can be an active observer within those things. Obviously, in real life, you wouldn't necessarily be in an IRB review or in a study section, but we think that there is a great potential for you to see these things in action and see how things are discussed. And people from the course who weren't involved in your trial development will be the ones who be in charge of reviewing your proposals on both of those ends. So in terms of the expectations, we're asking a lot of you, and there are a lot of things that we want you to accomplish, but we think that accomplishing these things within the milestones that we will set will be a recipe for success in getting your protocol done and then hopefully funded, knowing that there are vagaries of funding. But one of the nice things is, through the course, we've started to set up partnerships with some organizations that do grants for early phase investigations, and we have a partnership with the Neuromuscular Society, AANEM, and they will potentially, if there are meritorious proposals developed as part of this course, have a capacity to potentially fund some of those proposals. So doing the course and making the things happen when they are when we are asking you to, in order to and we'll set out what all the timelines and milestones are in the document we'll send around next week. But there will be sort of minimum requirements to get the completion certificate, and I think people who really hit all of the completion timelines extremely well, there's going to be versions that have distinction. This is not to say that if for some reason a new clinical trial comes out that sort of makes you have to completely revise and change your area of interest, or a sort of the pursuit of something that you want to have industry donate is not going to work out. We understand that sometimes there are forks in the road, and we are certainly happy and willing to be flexible about these things. But in general, we think prevention is the best strategy there. So a good detailed search of the literature and clinicaltrials.gov to make sure that the idea that you have isn't actually actively being done somewhere else already. These are the sorts of things that we hope to get in the early phase of the course. So again, we're going to have some expectations that we'll make more clear in the documents that we will send around, and we're going to try to make that as clear as possible for the whole year. So this should be hopefully very straightforward. And the idea here is not like is primarily to accelerate the chances that we think you can be successful in terms of getting your protocol done, and conceived in a scientifically good way, but also we think this will enhance the chances that it will be able to have an impact on science and the field that you're studying. So we're very near the end here, and we'll allow for some questions in a moment. So how excited are you for this? This is a loaded question. It's pretty much a push-pull because almost all the choices are pretty high. So yeah, if we were doing a survey design course, you could tell me that I'm basically terrible at coming up with surveys, and this is not a particularly valid survey. But okay, so people are pretty excited about this. I'm very excited about the group of you that we have managed to recruit for this year. Again, we did have a large number of applications, almost 2 for every spot. So I'm really excited about working with you all. And I think this is going to be a tremendous opportunity for us to help you move your science forward and also get really rigorous research into the pipeline for the clinical neurosciences. So let's see, I think I had one more. Oh yeah, so what are the next steps? So the instruments, you'll start receiving Qualtrics surveys from me and what's going to happen with those are not anonymous links. You will get reminders if you don't fill them out. And I basically, all your emails will be fed into Qualtrics and you will get them. Some of them will be batched, but some of them, we'll try to spread them out so you're not getting 3 emails a day from me. The small group pairing we're actively working on based on your availabilities. If there's anybody out there who hasn't sent us that spreadsheet that has the sort of matrix of times available, it would be great for us to have that as soon as possible. The general syllabus and the protocol for how we treat you in the course and what you're going to need to do when, we should be able to get to you sometime next week. I'm working on putting the finishing touches on that. I'll be discussing that with the other course leadership on Monday. So April is mostly going to be instruments and perhaps some recommended readings and video review. May will be really the early design activities with the small groups and also the attendance in the webinars. Again, if you're not able to physically be at the webinars, we do encourage you to go and review them, and we're going to try to get them posted as quickly as possible. And then in June, additional design activities through the small groups. And we're going to be working soon here to start to get those small groups in place, knowing that you're working with some busy people and that busy people sometimes have hard times finding times that are logical to meet. But we've done the first pass. We're starting to try to get the leaders into places, and we're going to fit the participants into places where the leaders are available. So at this point in time, I am now open to questions from the audience. Also, there are some resources on our website and a lot of the lectures from last year and a lot of the webinars from last year. But at this point in time, and obviously anybody can email us, probably the info email or my own email, which I'm sure you've received some emails from me before. So, it's totally reasonable to ask questions by email. But if anybody else has any questions right now, you can type them in the chat box, and I'll start to see if you're typing. And I won't belabor it if nobody has any questions, but I will use this opportunity to pause this and blow my nose, or I'm going to mute this and blow my nose. So hold on a second. Okay, so this cohort has 32 people, so it will be 8 small groups of 4. Concurrent to this, there is an advanced track that is tasked with developing some adaptive clinical trial designs, and we have 4 project teams there, and there are about 2 or 3 people in each of those project teams. But there would be very little overlap with that group. We will go out to dinner with them during July, but basically they're sort of freestanding courses for different populations with different goals. So that we have 32 right now. And that's what we have a wait list. So if there is any attrition, we do hope to figure that out soon and get some of the people from the wait list on there. But we have not had any attrition from our initial 32 selectees so far. Roger, did I miss anything? Roger is going to be largely involved in the conduct of the advanced track, but he's given some excellent discussions on DSMBs and IRBs and has vast experience in that area. So he will be somebody who you're going to be meeting during the course, along with obviously Chris and Lori and Robin. All right. Well, seeing that it's about 5 till. Oh, I see Roger is typing, so we'll let him put in his words. If anybody else, I can't hear you, but hold on. I might be able to. Let me see if I can. If you press, there's like a microphone button up at the top that you have to press. It's like a. We should have given you. As a presenter, you should have the ability to use audio. So if up in the top, you can press. Oh, we made him a host. So yeah, if you press the little button up at the top. Well, we probably should have done more of a rehearsal with Roger to make sure he's been. He is looking forward to meeting everybody, and sorry that we couldn't get his microphone to work there, but again, that is okay. Did anybody else have any questions from the group? All right, so stay tuned. We're going to be getting you a little bit more information in the sort of more omnibus protocol/syllabus document early next week. And we're going to send out some announcements about the rest of the webinars and work on getting those onto your calendar here soon. So it was a pleasure chatting with you all today, and I hope this was useful, and I hope it puts a little bit more clarity into what's going to be going on. And we'll be posting, if any of this was sort of faded out or you weren't able to catch all of it, we'll be posting this as a recording onto the website very soon. So thanks, everybody.